Hello everyone, my name is Pooja and I have completed my Masters in Medical Microbiology. From the past 1.5 years, I've been associated with academic writing and academic research. Today, we'll be discussing a very interesting topic in the field of microbiology, which is identification and isolation of bacteria from the mixed culture. So to begin with, we'll discuss what exactly microorganisms are. Microorganisms are basically pathogens which have the capacity to cause infections among human individuals. The identification and isolation of microorganisms involve basic steps like gram staining, biochemical reactions, and molecular level identification. However, the gram staining and biochemical reactions will not support to identify the microorganism at a species level. Therefore, the molecular level identification came into picture. These tests are significant to minimize the health burden by identifying pathogens. So, the healthcare professionals will be able to identify the pathogenic bacteria by following all these steps, including gram staining, biochemical, and molecular level identification. So the biochemical test is less reliable for identification of the pathogenic microbes as it supports identification up to the genus level, as we just discussed. So with the advancement in technologies, a new and advanced genetic-based test came into picture, which is also termed as molecular level identification. Although these tests, however, need skilled and trained practitioner, which is a common barrier in performing the molecular level testing. So, what is a procedure? We'll discuss in this slide, like what is a procedure that one can follow to identify the microorganisms from a mixed culture. A mixed culture is a culture which contains a lot number of bacteria, and every single cell is not isolated, and it contains a mixture of different pathogens. So there is a need to first isolate the specific bacteria from this mixed culture, and then to identify the bacteria to know the causative agent of any disease or illness. So the organisms from the mixed culture will be identified using serial dilution, gram staining, biochemical test, which is commonly known as IMVIC. What exactly IMVIC means, we'll discuss in further slides. It is followed by serial dilution, which will help us in performing molecular tests. The 16S ribosomal rRNA gene sequencing tool will be used to analyze the unknown organism from the mixed sample. This is essential as it will allow the healthcare professional or the technician to identify the bacteria up to genus level, up to species level, and just not based on the physiological properties of the bacteria. So this tool will help to detect the genetic material of the bacteria by relating to further its library or database for the final results. So what all are the materials and methods that are we going to use to perform these tests? Firstly, a mixed culture. Secondly, a nutrient plate agar. Nutrient plate agar is a basic medium where all of the bacteria can grow at a basic level, and from that culture the further growth can be proceeded, and further test can be performed to isolate the bacteria. This process will include various methods and instruments like sterile loops, glass rod, glass tubes, as well as incubator. Incubator is a system where we can incubate all the plates which are streaked with the microorganisms and will allow specific temperature for the specific growth of the microorganism. So these all are the materials in how we are going to perform the test. So next, the first step will include streaking. Streaking is a less sample is taken with the help of a loop and then streaked on a quadrant plate method. This diagram is clearly explaining how 4-quadrant means. The first quadrant will allow if a lot of culture. Further from the end of the first quadrant, the next streak will be done. This is known as second quadrant, and thereby third and the fourth. And in the other image, it is clearly known that by the end of the last quadrant, the isolated colonies we can procure, and from this isolated colonies further test can be performed. After every quadrant, there is a need to perform sterile and hot the loop, so that to avoid any cross contamination for the isolation of the bacteria. Further, this plate is incubated at 30 degrees Celsius, and a specific temperature is provided, and then after 24 hours the plates are recovered from the incubator and further tests are performed. All these actions and steps will include aseptic handling, which is necessary for controlling the spread of for controlling the cross contamination within the plate, so that isolated colonies can be procured from the mixed culture. So what is serial dilution? It is a stepwise dilution of a substance in solution. So we'll get a tube which is full of a sample, and from this there will be like 4 or 5 tubes. Every will contain an equal volume of solution, or the we can say or the agar medium, and from first tube will take 0.1 ml and then put it into the next tube, and from this tube will again take 1.1 ml and then transfer it to the third, and then to the fourth. In this way, the volume will remain same, however the concentration will decrease, so that the load of the bacteria will be less, and we'll be able to isolate the bacteria. This serial dilution is always performed by spread plate method. The sample which we used is 0.1 ml for every tube. So spread plating technique is a method of isolation and enumeration of microorganisms in a mixed culture and distributing it evenly. In this procedure, the mixed culture sample is spread with the help of a neutral plate with the help of a sterile glass rod. Sterile glass rod is basically a L-shaped glass rod which allows the complete distribution of the sample over the nutrient plate agar. This plate is incubated to 24 to 48 hours at 37 degrees Celsius in the incubator. This allows the perfect temperature for the multiplication of the bacteria and to isolate the bacteria. So this is how a spread plate technique looks like. Firstly, we'll take the sample on a nutrient agar plate, and then with the help of a sterile loop, with the help of a sterile spreader, we'll spread all the sample throughout the nutrient plate agar, and then we'll incubate it. Then after incubation to 24 to 48 hours, the colonies will be grown on the nutrient agar plate, and from this plate further tests can be performed. So apart from nutrient plate agar, there are other selective and differential plating methods which can be included to isolate a specific bacteria from the mixed culture. So apart from nutrient agar, differential agar, which is also known as blood agar. So in this presentation, all these steps will be based on a sample which will be provided to us, and from them we have to isolate a single bacteria. So on the blood plate agar, which is also a differential agar, in this agar plate, different organisms will show different hemolysis because the blood is present, which contains heme. So the bacteria, which is hemolytic, will show its hemolytic properties and thereby forms a zone of hemolysis. For example, the Bacillus cereus will show a beta hemolytic zone, which is specific to Bacillus cereus only. And the other is mannitol egg yolk polymyxin, also known as MYP agar. It is also a differential as well as a selective medium. For being a selective medium, it contains polymyxin, which only allows the growth of positive bacteria, and mannitol, which is a sugar, and the organisms which are able to ferment this mannitol will show pink colored colonies, and the one with which do not ferment this bacteria will show colorless colonies. And the third selective and differential media is EMB, which is also known as eosine methylene blue. It also contains lactose, and the colonies will be differentiated on the basis of lactose fermenting as well as non-lactose fermenting bacteria. These plates are further checked, and isolated colonies are further streaked on nutrient agar, and this is also incubated at 24 to 48 hours, and then further staining is performed. So these are the results which we got by by streaking our organism, which the organism or the sample hasn't provided to us, we will streak on those specific agar medium, and this is the result that we got. On nutrient agar, spreading and rough colonies were found. On MYP, smooth and pink color colonies and non-lactose fermenting, which indicates non-lactose fermenting. Blood agar, on blood agar, we got zone of beta hemolytic, which we just discussed that Bacillus cereus shows a beta hemolytic type of colony. And on EMD agar, small colorless colonies and differentiating lactose and non-lactose bacteria. So this is how we are going to perform gram staining. It basically contains 4 steps. In step 1, a smear will be prepared on the slide. All this procedure is being followed aseptically so that cross-contamination will be minimized. The first will include a crystal violet. Crystal violet is a primary stain wherein the bacteria containing a thick polypeptide and glycol layer or cell wall will imbibe this primary stain and gets fixed into the cell. And the other step is iodine. Iodine is also known as mordant. It makes the dye less soluble and so it adheres to cell walls. If the bacteria consumes or imbibes the primary stain, which is crystal violet, the mordant or iodine will help it to get fixed within the cells and do not leave the cells even after the third step, which is also known as decolorizing step. Decolorizes, washes away the stain from the gram cell walls, and in case the bacteria which does not imbibes the primary stain, which is crystal violet, will lose this color. And the fourth one is safranin. Those cells which lose the primary stain will gain the safranin, which shows a red color. So the gram positive bacteria will show a purple color cells. However, the gram negative bacteria, which retains the safranin color, will show the red color cells. So this is the way how gram negative and gram positive bacteria can be segregated, and will be able to further design a test based on the gram positive or the gram negative bacteria. So the biochemical test which will be followed in isolating this bacteria and the identification of this bacteria will be IMVIC. IMVIC stands for indole methyl red, Vox Proskauer indole citrate test, caclasses. These all are the definitions of IMVIC. So after going through this IMVIC test, the gram staining, as we just discussed in the previous slide, showed a gram positive color cells. It means the cells after microscopy will show a purple colored cell. And only few tests were showing positive, which includes caclasses, Vox Proskauer, citrate, and growth in KCN. These all tests, as well as the gram negative nature, will be counter-checked with the lab manual, and it was detected that all these properties are reflecting the suspected bacteria, which is Bacillus cereus. So how a molecular test will help us to go to a species level to perfectly understand what exactly is our identified organism. So after going through 16S RNA gene sequencing test, which is commonly used for identifying bacteria, because analysis of the organism DNA is often more definitive than classification based solely on phenotypic characteristics. So this will judge and identify the bacteria based on the genetic material and not solely on the phenotypic characteristics. So these are the details which we got after running the 16S RNA molecular test, wherein the query coverage was 100% and the identity with the identified organism, which was Bacillus cereus, was 96%. The accession number was AF176321. So to conclude, we can say that the identified bacteria was gram positive bacteria, which was clearly identified with the help of gram staining, and the other characteristics, biochemical characteristics, were identified with the help of IMVIC test, which also indicates the Bacillus bacteria. And further, with the help of 16S RNA, it was clear that the bacteria was Bacillus cereus. So the knowledge of microbiological techniques and aseptic techniques is essential in order to not misinterpret the results. The biochemical and molecular tests are more specific and sensitive for microbiological assays. The tools are also helpful to identify the microorganisms in epidemiological surveys at the time of outbreak for disease prevention and control. So this identification of bacteria in case of there is any outbreak or any epidemic, wherein it is essential to understand what is the identity and the characteristic of the microorganism, so that effective medications and therapy and treatment can be designed for the safety of the people at that time. So this technique and which includes identification of microorganisms is not only supporting the healthcare industry, but it is also more popular in today's food industry, wherein the different kinds of food pathogens can be detected at an early stage, and the food will be prepared with safety and for the safety of the consumers as well. So we can say that identification of bacteria is a very skillful technique, and the technicians must understand the need, and they must perform these tests aseptically in order to understand and identify the characteristics of the suspected bacteria, and with the help of molecular tests, the actual bacteria at a species level will be identified, so that the causative agent of the illness will be detected and the safety of the patients can be increased. So this all was about identification, how we can identify and the bacteria which can cause serious human diseases. So these all were the references which were used to design this presentation. So we can say that this presentation will surely increase the in-depth knowledge of the students who are facing troubles in identifying in the topics like identification of the microorganisms, and this will increase their knowledge how to isolate the microorganisms for the mixed culture as well as identify the organisms. Thank you and have a nice day.